welcome. Today we'll be discussing RNA and how RNA relates to and is made from DNA. So first, let's just start off comparing RNA and DNA. They're pretty similar in terms of overall structure. Couple key differences here. RNA is generally the copy of DNA because DNA cannot leave the nucleus. So anytime something is needed to be done outside the nucleus and a portion of the DNA is needed for that, a copy of the DNA is made, and generally that copy is RNA. In the backbone of RNA, it uses the sugar ribose instead of the sugar deoxyribose. If you remember, DNA molecules, the nucleotides are made of deoxyribose sugar, a phosphate group, and a nitrogen base. Same thing for RNA, except the sugar is ribose instead of deoxyribose. RNA is also single-stranded instead of double-stranded like DNA. So we can see that here, RNA single-stranded, DNA double-stranded. And then RNA contains the base uracil instead of thymine. So A would bond, or adenine would bond with uracil instead of bonding with thymine. There are three main types of RNA that you will need to know about. mRNA is messenger RNA, rRNA, also known as ribosomal RNA, and tRNA, or transfer RNA. So let's talk about each one of those here. The first one is messenger RNA. Messenger RNA is basically the copy of DNA that's going to be used to make a protein. So the DNA is stored in the nucleus. We can see that here. DNA is like storage if we're comparing this to computer and programs and stuff like that. The mRNA would be equivalent to like a software, which includes the set of directions, and it comes directly from the DNA here. And then you would use this software to make applications or to do certain functions. And the end result, that's proteins, right? So DNA is the original thing, RNA is the copy, the whole goal is to make a protein at the end. And that's what mRNA is. mRNA is that copy of DNA. rRNA is a little bit different. rRNA makes up part of the ribosome. The ribosome is a cell organelle that's main job is to make proteins. So there are two subunits of a ribosome, a large subunit and a small subunit. Each subunit consists of several different rRNA molecules kind of linked together, and each ribosome can make up to as many as 80 different proteins. And then this thing running through it is actually the mRNA. And then finally, we have tRNA, which is known as transfer RNA. Transfer RNA's main job is to carry amino acids from the cytoplasm to the growing polypeptide chain. So what happens here is just kind of putting this all together for you here. Here's a ribosome. The ribosome is trying to make a protein right now. What's being fed through the ribosome is the messenger RNA. And then these little molecules carrying amino acids right here are called tRNA molecules. The tRNA molecules, their job is to carry amino acids to the ribosome so that those amino acids can be used to make a growing polypeptide chain, which will eventually turn into a protein. So three types of RNA there: mRNA, rRNA, and tRNA. I bring that up because you need to know about all three types of RNA as we go through this process of protein synthesis, of how you make a protein. This process of going from DNA to RNA to protein is called the central dogma of molecular biology. It's how everything is made in your body. Anytime something needs to be made, a portion of DNA is copied. That DNA that's copied is RNA, usually gets edited, and then that edited RNA goes to make a protein. So that process takes place in two different steps. The first step is transcription. That's what we're talking about today. Next video, we'll discuss translation. So transcription is when the strand of DNA is copied into a strand of mRNA. Translation is then when the mRNA is used along with ribosomes and tRNA to make a polypeptide. So here's step one, right, of protein synthesis, DNA to RNA. So it's got steps. If you remember the steps of replication, it's pretty similar. Right? First step of replication is DNA unzips. Similar here, DNA unzips. Then you have RNA polymerase attaches to the DNA strand and pairs up free nucleotides, similar to how DNA polymerase would have worked. But RNA polymerase makes RNA instead of DNA. And then a new backbone is formed on the end of it with sugar and phosphate. So something we can do is we can practice doing transcription, turning a DNA sequence into RNA. And it works pretty similar to how replication would have worked when we were trying to figure this out here. So if I have one strand of DNA on your screen, you should be able to figure out the complementary strand, right? T bonds with A, but then a key thing is there's no T on RNA. So where we would put the T in the second space here, instead there's a uracil. You can see how that looks here, right? So T goes with A, but A instead of going with T goes with U because this new red strand here is mRNA that's being made, and RNA doesn't have thymine. Right? C still goes with G, A goes with U, but T still goes with A, right? So that's a little bit of practice, and we're going to go through and work on this today once we're done with our notes. So that's how the cell goes through, copies DNA to make an mRNA molecule here, right? So we have a long string of mRNA. The next step is that the RNA needs to be edited. This is the unedited version of the RNA, but oftentimes the DNA has built-in sequences that are basically just junk DNA that doesn't code for a specific gene. That DNA does have certain functions, like it helps to protect in case of mutations. Like if there's a mutation, there's a chance it could happen in this junk DNA and not affect the actual gene. But that junk DNA needs to be removed, or that sequence needs to be removed from the mRNA. And so we have different parts of the RNA called introns and exons. The introns are the copies of the DNA that didn't code for anything, and the exons are the parts that actually code for a gene. And so what happens are the introns are removed, like we see in this picture here, and then the exons are spliced together until we get a mature RNA sequence. And that mature RNA sequence would then leave the nucleus, go into the cytoplasm, meet up with a ribosome, and start to make a protein in the next step, which is translation.